Hi, I'm Sam Mosseri, and I will be going over a study which investigated the crystal structure of the GLUT1 protein. So, what is GLUT1? GLUT1 is an integral membrane glycoprotein, which is encoded by the gene SLC2A1 and contains 492 amino acid residues. Uh, as you can see here, here's the GLUT1 uh, glycoprotein in the membrane of an endothelial cell. It's responsible for the cellular uptake of glucose. It's not the only way, but it's an important way for erythrocytes and endothelial cells of the blood-brain barrier specifically. Also other organs as well, but those are the two very important um, cells which utilize GLUT1. As a brief word on the mechanism, it's a uniporter, so it only transports one molecule of D-glucose. The D-glucose binds to the active site, which induces a conformational change, which is energetically favorable, and it is therefore able to transport glucose down its concentration membrane, its concentration gradient, um, through facilitated diffusion. So it does not require ATP. It's part of the major facilitator superfamily. Given its important physiological role, you can imagine that there would be some diseases associated with it, and there are. Overexpression of GLUT1 is found in many cancer patients. Additionally, GLUT1 deficiency syndrome is caused by loss of function of the GLUT1 proteins. This is a caused by genetic mutation. And it's a very rare disorder. It's only 500 confirmed cases have been known since the 1990s. And symptoms of this neurological disease include frequent epilepsy, microcephaly, dyskinesia, involuntary muscular contractions and movements, and developmental and intellectual disabilities. And these symptoms start manifesting themselves during infancy. And then also it's possibly related to, GLUT1 is possibly related to just epilepsy in general, although its association with that is somewhat unknown at the moment. So given its role in disease and physiology, it'd be very useful to have a 3D structure, a conformation of this protein. And so that was the overall goal of the study, was to crystallize GLUT1 and map the three-dimensional conformation. And it was successful. Now the initial attempts were unsuccessful, and here just a brief word on the methods. They were unsuccessful, it's thought, because of the frequent changes in conformation. Really, it really prevented successful crystallization. However, after searching the literature, they were able to find a point mutation, E329Q, which locked the protein in an inward-facing conformation. And after that, they were able to successfully crystallize it. So that was really a key step in the success of this experiment. As far as the results, here's the general structure. It's a 9 to 455 residue fold, which is just common to the major facilitator superfamily. And they divided it into 3 domains. Here's the N domain here on the right, C domain on the left. These are transmembrane domains, which are divided into 12 transmembrane segments total. Then there's an intercellular helical bundle domain here in the intracellular side, which this ICH domain connects the N and the C domain. And interestingly, they seem to be unique to sugar porters. So that's an interesting piece there. As far as implications for ligand binding, here there's outward open conformation, which in which the ICH connects the N and the C domains, and then in the absence of the ligand, the sugar, when the sugar binds to the active site here in the C domain, it induces some conformational shift in which the 7 and 1 transmembrane segments start interacting, and you have the simultaneous dissociation of the ICH domain during the secluded state. And here with the inward open conformation, you have this full association between the 7 and 1 transmembrane segments signified by this little red brick. And then the glucose is here exposed to the intracellular environment and will dissociate if the concentration is low enough. Once it dissociates, the protein reverts back to its original outward open conformation, which is energetically favorable due to the strong interactions of the ICH domain. Now, a really important part of this study was the ability to map out disease-associated mutations. And they mapped out 3 clusters. Cluster 1 involved mutations which affected the substrate binding site, so the active site here. Cluster 2 involved mutations in the ICH domain, which would prevent the successful latching in the outward open conformation. And these were accounted for about half of the disease-associated mutations. Cluster 3 involved mutations in the extracellular gate, which would prevent a proper intracellular open, inward open conformation. So here on the next slide, they have a table. These disease-associated mutations were already known before, and here they're associated with certain diseases, GLUT1 deficiency syndrome 1 and 2, and then just general epilepsy. So what's really useful though is that now each one of these mutations is able to be mapped into these clusters, which give an indication of how these mutations mechanistically affect the dysfunction of the GLUT1 protein. So that just overall gives a general overview of the study, and it provides a platform for future research. Thank you.